This is the Bio 104 PowerPoint for Lab 8 on photosynthesis. The important thing for lab is knowing the overall equation for photosynthesis. This is how we're going to evaluate if this process is occurring or not. So knowing the reactants and the products allows you to determine if the process is occurring. So you need to know that the reactants involved in photosynthesis are carbon dioxide, water, light energy, which will be used to produce glucose and the waste product of oxygen gas. Overall, photosynthesis is converting light energy and storing that energy as sugar. And this occurs in the chloroplasts in eukaryotic cells, which make up the structure of plants. Plants also have stomata, which are basically pores, which allow different gases to enter and leave the plant structure. In the process of photosynthesis, carbon dioxide is entering, oxygen gas and water vapor are leaving. Also during photosynthesis, you have oxidation and reduction. Oxidation is the loss of electrons, water will be oxidized, and reduction is the gain of electrons, and the carbon dioxide will be reduced. This is a picture of the overall process of photosynthesis. It involves two steps, the light-dependent reactions and the light-independent reactions, or the Calvin cycle. And this also shows you how the reactants and the products are involved. So remember, for photosynthesis to occur, you need water and carbon dioxide and light energy, and this will go through the two processes to generate oxygen gas as a waste product and sugar. So a little bit more detail on the two different processes. First are the light-dependent reactions. Light-dependent reactions, these occur in the thylakoids. Thylakoids are those green membranes that you see in the chloroplast, stacks of membranes. Chlorophyll and other pigments are embedded in these membranes, and their job is to absorb the light energy. And then this light-dependent reactions will convert that light energy into chemical energy, and those chemical molecules are ATP and NADPH. And there are two photosystems. This is an image of the two photosystems. Photosystem 1 occurs second, and that is the NADPH producing photosystem. So that is going to produce the NADPH. The second photosystem actually occurs first, and that's the water splitting photosystem. And that is going to take the water, which is a reactant, and split it, break it apart to produce the oxygen gas. And of course, these are both photosystems, so they both involve light energy. The second part of photosynthesis is the Calvin cycle. Calvin cycle is sometimes called the light-independent reactions because light is not required. So light-independent reactions. And this occurs in the stroma, which is the liquid part of the chloroplast. And this involves the carbon dioxide and the light-dependent reaction products, so the ATP and the NADPH. And what they will do is produce the glucose. So this is an image of the Calvin cycle, and again, it requires the carbon dioxide. It also uses the products of the light-dependent reactions, the ATP, the NADPH, and you go through a cycle to generate glucose. So again, a review of the two parts of photosynthesis. So light-dependent reactions, light-independent reactions, the Calvin cycle, and the reactants, you need water and carbon dioxide and light energy. The water and light energy are required for the light-dependent reactions and will result in the production of oxygen. Carbon dioxide is required for the Calvin cycle, as are the ATP and NADPH from the light-dependent reactions, and they're used to produce glucose. All right, for your experiments, I'm going to go over the experiments, the procedures, and the data that you need to record for your lab report and to turn in. So experiment one is the algal ball experiment. And one of the things you need to do, would have done to prepare for this experiment, is to take some water and added a pH indicator to it. The original color would have been this purple color. Then you would have put a straw into the beaker and would have blown CO2 into the solution. And as you blew the CO2 into solution, the solution would have turned yellow. So this indicates that the pH of the solution was decreasing or decreased. So you would have prepared that solution. Second thing you would have done is to prepare the algae balls. So to prepare the algae balls, you would have mixed algae with some other solutions to form these little balls, sort of like jelly balls of algae. After forming the balls, you would have rinsed them, drained them in a strainer and rinsed them, and then you would set up the vials for the experiment. All right, the vials are very small containers, and you would have filled them with that yellow solution that you had produced that has high levels of carbon dioxide, and you would have put in the little algae balls. So you would have prepared 2 vials that would have looked identical. Then one vial would have been covered in aluminum foil. So you'd have completely covered the vial with aluminum foil, which will prevent light from getting in. So you would have had one vial covered with aluminum foil, the other vial would have left open, and both of those would have been put under UV lights, grow lights, for a period of time. When the experiment is done, you would have vials that were covered by the aluminum foil. The color would be yellow. So this indicates they were not exposed to light, no light. And the vials that were exposed to light would have changed color back to the original sort of purple color. So these were exposed to light. So this is the data that you need to record in your lab. So again, the vials that were covered by the aluminum foil, the solution would have remained yellow. The vials that were exposed to the light, the solution would have turned back to the original color. So that's the data you need for experiment 1. Experiment 2, this is the leaf disc assay. So for this, you would have taken spinach leaves and cut out little discs, little round pieces of the spinach leaf. Then you would have taken those discs and put them into a solution and infiltrated them with the solution. So first solution you could have used was tap water, and you would have put the discs into a syringe, filled it with tap water, then sealed the top, put your finger over the top, and then pulled down on the plunger to create a vacuum. And what this would have done is it would have forced the liquid into the leaves and forced out the gas. So the gas would have been removed. And this would have caused the leaf disc to sink to the bottom of the solution. So you were replacing the gas that was in the leaf with the liquid. After you had done that, you would have put those infiltrated leaf discs into a beaker with the solution, and all the leaf discs should be sinking to the bottom because they have been filled with liquid, which is more dense than gas. So what you would have done is do this procedure using a bicarbonate solution, and then you would have had a separate group of leaf discs where you would have infiltrated with the aged tap water. You would have put both of those beakers under the grow light and then check them every 5 minutes. And you would check them to see how many leaf discs were floating. So this is the set of data that you should use for your experiment. So the time points are every 5 minutes, 5, 10, 15, and so on. And I've recorded how many leaf discs should have been floating at each time point. So at 5 minutes, in the bicarbonate solution, you would have had 3 discs, and in the aged tap water, you would have had 0. This is the data you should use, and also in your lab report, you are going to be making a graph. So as a review to making a graph, you need 2 axes, a y-axis and an x-axis. You need a title for your graph. You also need to label the axes, so you need to label here and label here. And when you add the data, you want your data to fill the whole graph. So whatever your data line is, it should fill the whole graph. You don't want your data to just be in a small part of the graph. All right, so you should have all the data for experiments 1 and 2 and be able to fill out your lab report. Make sure that you write really neatly so that I can easily read what your answers are.